Good morning. My name is Vipul Jarath. I'm Vice President of Medical Research and Development here at Alimentiv, and I'm Professor of Medicine at Western University in Canada and practicing gastroenterologist. Following our launch event earlier this year, I'm very pleased that more than 800 people have taken time out of their busy schedules to join us today here for our second live stream event. I believe this high level of interest reflects the topic and, of course, the speakers. Today, patients who suffer with inflammatory bowel disease have several therapeutic options available to them, and no doubt there will be many more on the horizons given the number of drugs that there are in clinical development at this time. That said, our approach to treatment is largely based upon trial and error. We have very few tools in our toolbox to really reliably identify which patients are most likely to respond to treatment and really enable us to pick the right drug for the right patient at the right time. I think until a time we have treatments with much greater efficacy than we do today, a more personalized approach to treatment really must hold promise to improve this situation. Our topic today is precision medicine in inflammatory bowel disease. We're fortunate to be joined by some of the top experts in our field, Dr. Séverine Vermeer, Dr. Diana Nguyen, and Dr. Niels van der Kasteel, all individuals whose visions and efforts have helped move this field forwards. Just some housekeeping for today's presentation. Our keynote presentations will be followed by a panel discussion for 15 minutes. All the lines are muted. However, you're very welcome and you're invited to submit questions using the chat box that you'll see on your screens, and you can do this throughout the presentation. We will select questions for the panel from these, and any questions that we don't have time to address due to restrictions, we'll endeavour to answer these subsequently by email. We have a full agenda for the next hour, so let's get started. Well, I'd like to introduce our first speaker, Professor Séverine Vermeer. To those who are in the field of IBD, she needs little introduction. She's one of the foremost experts in our field. By background, Séverine obtained her MD degree at KU Leuven in 1995, and her PhD was on genetic polymorphisms and serological markers in IBD. She did further training at the Wellcome Trust for Human Genetics in Oxford and then McGill, and since 2003, she's been a staff member of the gastroenterology department at the University Hospitals Leuven. She's a full professor of medicine at KU Leuven, and she heads the Department of Chronic Diseases, Metabolism and Aging. Dr. Vermeer has been a principal investigator in multiple randomized trials of new therapeutic compounds, and she's led some of the development programs, many of those, in fact. Her scientific works resulted in more than 600 peer-reviewed articles, many of which focus on identifying predictive signatures of treatment response. She was president of European Crohn's and Colitis Organization from 2014 to 15, and a previous president of the Belgian IBD Research Group. Séverine joined us today to talk about how precision medicine can advance clinical research and clinical practice. Séverine, welcome and thank you for doing this, and the floor is all yours.
Thank you very much, Vip. Good evening, everybody. Thank you also for the very kind invitation and the kind words. So I have some disclosures. And I'd like to start by showing this slide. This is a slide I compiled many years ago, but I think it remains still very up to date to our discussion. These are all patients with inflammatory bowel disease. So in fact, they all have the same disease, or maybe two diseases, Crohn's disease and ulcerative colitis. But I think the pictures are showing you that there is substantial heterogeneity. Now, even if we focus only on the luminal side of the disease, these are 4 different patients with Crohn's disease, and you see that there is inflammation in all of these patients. However, as clinicians, we know from clinical practice that not all patients need to be treated in the same aggressive manner. There is also inter-individual heterogeneity in disease course. It's very well described that at the time of diagnosis, 90% of patients with Crohn's disease will fall into the inflammatory behavioral subtype. When you follow up patients over time, then after 5 to 10 years, 25% only will remain in this inflammatory B1 subtype. A third of the patients will have evolved towards stricturing or stenosing disease. a third will have evolved towards penetrating, fistulizing disease, and probably 5% will have developed very fulminant and multi-refractory disease. So we are really in need, first of all, to identify who needs a more aggressive approach to the disease. And I think the good news is that we now have expanding treatment options beyond our 5-ASA, steroids, and thiopurines. We can now target in a more or less specific way specific cytokines or pathways. We can target the migration of specific cell types. However, I think if we look at all of these agents, they are all anti-inflammatory. They tackle, in fact, only the inflammation. I think we have to dare to admit that none of these agents are tackling fibrosis. None is tackling the microbiome or the barriers specifically. So I think there's still some progress to be done. So as clinicians, again, moving back to these 4 patients, we want to achieve with our current agents clinical remission, no steroids, and some endoscopic improvement or healing at least. But it's very often trial and error. We try a drug, our drugs have equal efficacy. However, there's still a problem. When you look at, this is the example I give you for Crohn's disease, an endpoint of clinical remission at week 52, and you see the 4 registrational studies for infliximab, for adalimumab, vedolizumab, and ustekinumab. What we know is that there are patients who are primary non-responders, and second, there is a therapeutic ceiling. We seem not to be able to overcome the 30-40% clinical remission and endoscopic improvement rates. And this is, I think, still a problem. There's another complexity, and this is that there is a time-dependent variability of the biological responses in IBD. And I think that variability dictates also the need for different therapies. So while a genetic susceptibility, together with some life events that may alter, for instance, a methylation profile, this will render an individual into a state of physiological inflammation. However, when there is an additional trigger, which is often, very often unknown, that will really lead to onset of disease, where maybe one pathway is predominantly overexpressed. But then during the life of a patient, there might be a viral infection, or the patient might resume smoking, or there might be a bacterial infection. And all of these events might lead to yet different pathways that are being disturbed, overactivated, dysregulated, and most likely all of these events might also require a different approach or a different start of a therapy. And all of this complexity at the moment, we're absolutely not taking into account. So this individual variability in our genetic background, in the environment, in the lifestyle of a person, I think needs to be taken into account when we approach also the management of disease. And this is what precision medicine really tries to do. That the genetic susceptibility towards disease is not the same as disease prognosis was shown by the UK, the group in the UK that reanalyzed their genome-wide association scan, and they reanalyzed the GWAS by prognosis. So they compared 1,700 patients with a poor prognosis to almost 1,000 patients with a good prognosis. And they identified by reanalyzing their genome-wide association scan that 4 loci met genome-wide significance. And over the years, the group in Cambridge, led by Ken Smith, but also Miles Sparks and James Lee, further worked on trying to identify in the peripheral blood a signature that could identify patients with a more or a less aggressive disease course. And after cell purification and sorting, they identified a CD8 T-cell signature and a signature of T-cell exhaustion, where they showed that the more T-cell exhaustion there was, the more mild the disease was. The interesting thing was that this was not only shown for inflammatory bowel disease, they really replicated their findings over the past years also for other inflammatory disorders, including vasculitis, pulmonary fibrosis, and lupus. So interesting findings. of something that we can find in the blood of patients. Now, if you have to sort cells, it's of course not practical. So they have then moved on in examining if the same signature could also be traced back in whole blood without cell sorting. And they eventually, after very extensive validation, identified a 17-gene qPCR classifier, so a test which has now been commercialized and which is to be measured in the peripheral blood of patients. And so this prognostic panel of markers, I would call, is now being tested in a prospective study, which is called the PROFILE trial. This trial is well underway in its recruitment. And so these are patients with almost newly diagnosed active Crohn's disease, where the biomarker will be assessed. Patients will be either put in the IBD high or the IBD low group, so patients with a bad prognosis versus the patients with a more mild disease. And then in each of these two categories, patients will be randomized. And what you, of course, hope to show that in the IBD high group, so these are the patients that are very likely to progress and to have a more aggressive disease course, that a top-down strategy would be more beneficial in these patients and then could revert the disease. The primary endpoint is sustained steroid and surgery-free remission. So I think a very important milestone study that hopefully will give us the answer whether it will be feasible to subgroup patients at least. Now, in the meanwhile, as clinicians, we don't have molecular markers. So what are we doing in clinical practice? We use clinical features. We use features of disease subphenotype, comorbidity, severity, side effects, the preference of the patient, and all of these more soft things, I would say, are used in helping us in deciding which drug we will use for which patient. And when we see these three, I would say, very rare phenotypes, but also very aggressive phenotypes, I think these are the rare phenotypes where, for clinicians, it's rather easy to decide. Because I think for very acute, severe UC in the middle or to the right, extensive perianal disease, here most physicians would go first line for intensified infliximab in combination with azathioprine, because specific dedicated studies have looked into these rare phenotypes. However, again, I want to emphasize this is rare, and this is not something, fortunately, we are encountering every day in practice. So for the vast majority of our patients who do not fall into this rare category, after identifying, for instance, by using a marker such as the Predict IBD Sure, that tells us who is the patient in need for more rapid escalation to advanced therapies, we would like to know as clinicians, in the middle, all the individuals in different colors, but we would like to know these are the patients we have identified, they would need more advanced therapy. But who will benefit most from drug B or drug C? And for this now, many of us are using different omics layers. We use proteomics, we use genomics, we use transcriptomics, we look at the microbiome in trying to find subgroups of patients. However, you need to know that behind each of these complex systems, there is a network. And the network really defines the interaction between the different components. And this is what we also need to take into account. And it's a little bit like we tonight, all of you who are listening, some of you are probably doctors, some of you are scientists, biologists, there are probably also students. And at this moment, you're listening to me, to the speaker. Now, if I would not have turned up this evening, there would not have been, at least for the first part of the webinar, a speaker. And this is what would happen. Our whole network would fall apart. So this is a little bit what people are trying to do, identifying the networks, the integration, and see which central node is controlling the network. And so people are starting to integrate the different omics layers to identify disease networks and the hubs that control it. It's easy to combine or to integrate just two clinical features with, for instance, the genome. But then you can do the same thing with the epigenome or with the transcriptome. And each of these interactions will identify a regulatory network with controlling hubs. And at the end, the hope is to identify really a disease network. So we tried some years ago by means of integrating clinical features with bulk transcriptomics in biopsies from patients that were about to start infliximab. And we identified a 5-gene signature that predicts response or non-response, depends on how you see it, to infliximab in colitis. One of the key genes was interleukin receptor alpha 2. We did some further experiments and studies in the last years when we knock out interleukin 13 receptor alpha 2 in mice. These mice have the same disease severity. However, they more rapidly recover after chronic DSS. We also identified in the biopsies a high expression of interleukin receptor alpha 2 in epithelial cells during inflammation. So we hypothesized that this molecule really has a negative influence on goblet cell recovery and the function and on epithelial restoration. We showed recently that the signal is anti-TNF specific, that we observed it also for adalimumab as for infliximab, not really for vedolizumab. Likewise, the group around Fiona Powry and Nathan West some years ago identified oncostatin M and the receptor to be implicated in or as a predictor of non-response to anti-TNF in patients with colitis. So in the left panel, you see that the levels of oncostatin were increased before treatment. They confirmed their findings in 2 phase 3 randomized controlled trials with infliximab and with golimumab. So pretty strong data. And in fact, this compound is also to be taken up in a study that where oncostatin will be blocked in patients with Crohn's disease, which is about to start soon. And maybe as a last example, TREM1, the triggering receptor expressed on myeloid cells, which is really an amplifier of the pro-inflammatory innate immune response. And this was identified by an Israeli group and also by ourselves in Leuven. What is puzzling us still is that we find opposite effects, but clearly TREM1 plays a role. It differentiates the responders from the non-responders. When you look at its function in stimulating, I think, immune responses, also in atherosclerosis and in accumulation of lipids in cardiovascular diseases, it would make sense that higher levels would be associated with non-response. And in a recent study, in a collaboration between Cincinnati and Israel, also TREM and oncostatin were identified to be involved in the network that is controlling really response not only to anti-TNF, but also to vedolizumab, to integrin agents, and also to glucocorticosteroids. So I think we're starting to identify some central hubs that control specific networks and that are associated or predictive of therapeutic outcome. Likewise, like we're trying to identify predictors of therapeutic outcome, predictors of prognosis, people are also trying to identify predictors of adverse events. And there, the UK group, led by Tariq Ahmad, is doing some beautiful work and identified, for instance, specific haplotypes that are associated with pancreatitis to thiopurines, but also in the PANS cohort, they identified HLA haplotypes that are associated with developing antibodies to anti-TNF, not only infliximab, also adalimumab. And in fact, 18% of the variance in immunogenicity to anti-TNF was explained by this haplotype. So little by little, we are starting to identify markers that I believe should be added or given to the clinicians to test in their practice. And likewise, some years ago, when we tried to see who were the patients who develop anti-TNF induced paradoxical psoriasiform skin lesions, we use a combination of clinical features, autoantibodies, and a genetic risk score that looks at psoriasis and atopic dermatitis genes. And you can see when you start building these into matrix models that you can predict if a patient is really at low risk to the left bottom, or if a patient is at higher risk when he or she has a higher genetic risk score, is starting infliximab at a low age, and is double-stranded DNA positive. So this is the kind of studies people are doing. I think, and I'm coming towards the end, that it's critically important that the same patient omics data integration is done for identification of relevant networks. So to the left is one individual patient with different omics layers where we want to integrate them and come to an individual, for that patient, individual IBD network. And you do the same with the patient to the right, and that patient will have her own individual IBD network. So on the top of the slide, you see all individual patients. And if you do that for a large number of individual patients with the same omics data, then you could start pool data and identify, I think, disease networks. And some patients will fall into the same network. We can, by doing so, we can identify molecular subgroups of patients. In this way, we hope, of course, to identify biomarkers. We hope it will also help us in precision therapy, therapeutic outcome markers, and eventually also in maybe drug testing, which should be done on a more homogeneous level as what we are doing now. We're just lumping patients together. We think they all have Crohn's disease, but they're so heterogeneous. So imagine that if you can do this, if you can integrate all the different omics layers, and you can identify a central regulator of a specific network, and you could develop or have a small molecule which specifically inhibits this regulator, then you can construct much more smart studies that only target this specific patient population who carries this central regulator. So to everybody who's listening, my conclusions, and I'll show everything maybe all together. I really think that prognostic and therapeutic biomarkers are more than ever needed to make progress. I hope I convinced you of that. I think on the clinical side, for us clinicians, we still can improve on some harmonization of definitions. It's crucial to validate. We cannot do this on our own. We need consortia, we need big cohorts. We need a good bioinformatic pipeline, but I think we have so many good, excellent people around the globe that can do this integration. But I do believe that integrating patient omics data is the way forward for grouping of patients, for biomarker discovery, for precision therapy, and for drug testing. And with this, I would like to thank my whole team and also you for your attention. Thank you.
Okay, great. Many thanks, Severine, for that really fantastic overview. And I think just in the interest of time, we'll go on straight to introducing our second speaker now. So I'm very pleased to introduce our second guest, Dr. Diana Nguyen from Theravance Biopharma Inc. Diana is a gastroenterologist, and she's Senior Director of Clinical Development at Theravance Biopharma, and she's the clinical lead for their drug development program in gastrointestinal disease. Prior to joining Theravance in 2015, Diana was a physician scientist, and she was an NIH-funded mucosal immunologist at the Massachusetts General Hospital in Boston. And her work has really been on the topic of IBD for the last 20 years. Prior to being on faculty at MGH, she had an appointment at Harvard Medical School as an assistant professor, and she completed all of her undergraduate and postgraduate medical training and advanced IBD fellowships at the MGH. Even in her current role, she continues to see patients with IBD at the San Francisco VA. Today, Diana will share with us how a translational medicine program was able to successfully support the development of their current compound, which you'll hear about, a gut-selective pan-Janus kinase inhibitor. Diana, again, thank you for doing this, and welcome, and the floor is over to you.
Thanks for the introduction. Good morning, good afternoon, good evening, wherever you are in the world. So over the next several minutes, I would like to tell you the story of TD1473 as an example of how to bring a new concept of an oral gut-selective molecule from concept through preclinical testing to early development. These are my disclosures. So as background, TD1473 is an oral, once daily, gut-selective pan-JAK inhibitor. And I'll explain what that means in the next slide. TD1473 is being developed for the treatment of Crohn's disease and ulcerative colitis. And most of the data that I'm going to show you over the next several minutes have recently been published in the September issue of the Journal of Crohn's and Colitis. Before I delve into TD1473, I would like to remind you of what a JAK inhibitor is. As you remember, there are high levels of pro-inflammatory factors called cytokines in the intestine of a patient with IBD. And these cytokines serve as signals from one cell to another that there should be inflammation. And when the cytokine, which is depicted in red on the left, docks on a receptor, which is depicted in blue, on the surface of a cell that is supposed to receive that inflammatory signal, it initiates a cascade of events, starting with the activation of an enzyme called Janus kinase, or JAK. An activation of JAK then activates further downstream proteins, such as STAT, which then goes into the nucleus and starts a cascade of events leading to further inflammation. So, as you can imagine, a JAK inhibitor can serve as a brake on inflammation, as evidenced by the positive phase 3 data with tofacitinib in ulcerative colitis. Similarly, there have been positive phase 2 data in Crohn's disease with filgotinib and upadacitinib, other JAK inhibitors. All these inhibitors that I've just mentioned are systemically active. And systemically active JAK inhibitors could lead to adverse events such as anemia and infections, including opportunistic and serious infections, due to the fact that JAK is also crucial in important cellular processes, such as the production of red blood cells and immune cells. This is where TD1473 is different. It is not systemically active, but rather it's gut selective, which means that most of the molecule stays in the intestine and does not get absorbed. Therefore, the plasma concentration in the systemic circulation is low. And from this, we hope this will lead to low risk for systemic adverse events. Therefore, a higher dose may be used, and therefore gaining efficacy due to the higher dose. So the idea of an oral gut-selective compound, such as TD1473, is to maximize therapeutic index by maximizing efficacy and minimizing safety concerns. So the gut selectivity was first demonstrated in animal models. Here you can see in a graph in rats that for a dose of TD1473 that yielded similar intestinal concentrations of drug, that led to a very low plasma drug exposure with TD1473 compared to tofacitinib. So in the graph, you can see that the GI selectivity is more than 3 orders of magnitude on a log scale lower with TD1473 than tofacitinib. And it's likely due to this gut selectivity that we found that the dose leading to a similar reduction in disease activity in this mouse model of colitis shown on the left resulted with a lower dose of TD1473 than tofacitinib. So that's efficacy. In terms of safety on the right, when we tested a very, very high dose of TD1473, in fact, 100 times the therapeutic dose that you see on the left, despite that high dose, we did not observe a similar reduction in the number of natural killer cells, which are immune cells, in the spleen of these mice. So taken together from the left and the right, the gut selectivity led to efficacy with a lower dose of TD1473 without a sign of systemic immunosuppression, even at high doses of TD1473. So with those encouraging preclinical data, we then took TD1473 into the clinic. I don't have time to show it today, but the healthy volunteer study showed that TD1473 was generally well tolerated at high doses with low plasma concentrations, the way TD1473 was designed. Over the next several minutes, I will then describe the results of our subsequent study, which was a small exploratory phase 1b study looking at the effects of TD1473 compared to placebo with 4 weeks of treatment in patients with active UC. Now, the main objectives of this small study was to confirm the benign safety and PK profiles of this compound in patients with active UC compared to healthy volunteers. However, we also looked at the usual outcomes of a UC study, even though this was not a full-blown induction study and was not powered for those usual endpoints. So these were exploratory endpoints. So 40 subjects with moderately to severely active UC, as evidenced by the presence of diarrhea and rectal bleeding, and an endoscopic subscore of at least 2, were randomized to 4 weeks of placebo or TD1473 at 20, 80, or to 70 milligrams. There were study visits at week 2 and week 4, which every subject had a full evaluation of efficacy and safety, including centrally read endoscopy and biopsies. After week 4, study drug was discontinued and subjects came back 2 weeks later off study drug for a final safety evaluation. So in terms of pharmacokinetic data, once again, we saw gut selectivity even in patients with diseased colons. On the left, you see a graph of the plasma concentration at different time points after the dose was administered at the week 2 visit. You can see that at all 3 doses of TD1473 depicted in the color lines, the plasma concentrations in these groups were very low compared to the dotted black line, which reflects what would have been seen with tofacitinib 10 milligrams BID extrapolated from published literature. So you can see that even at a high dose of 270 milligrams of TD1473 once daily, the plasma concentrations were at least 10-fold lower than what would have been seen with tofacitinib. So that's plasma concentration. In terms of colonic tissue concentration, you can see on the right that there were high levels in the sigmoid and rectum. And in fact, we had to graph this data on the right using a log scale so that all the data points would fit in the slide. And you can see at 80 and 270 milligrams, the colonic tissue concentrations were in range of what would be expected to be adequate to inhibit all the different pairings of the JAK subtypes. And this graph likely is an underestimation of the real colonic tissue concentrations due to the 2 enemas that each subject had to use as bowel prep before the endoscopy. Not shown on the left, but the plasma concentrations of TD1473 were below the concentration that we required to inhibit the JAK enzymes at 50%. So this really shows that the gut selectivity is still in effect in patients with active UC. In terms of efficacy, we found that TD1473 at all 3 doses led to greater reductions in C-reactive protein compared to placebo at all the time points examined during study drug administration, which were week 2 and week 4. So here is a graph showing the mean plus and minus 95% confidence interval of the reduction in CRP of each TD1473 dose relative to placebo, meaning that the effect of placebo had already been subtracted out. So there are several things I want to point out here. First is that you see that all the triangles, meaning the mean CRP reduction of all doses at week 2 and week 4 lie to the left of 1. This means that the reduction that was seen in TD1473 groups were greater than the reduction seen in placebo. Second is that this greater reduction is already seen at week 2, which is the first time that we measured CRP after study drug started. And then this greater reduction then was enhanced at week 4. And after study drug was discontinued at week 4, the CRP levels started to come back to baseline at week 6, the day 482 visit. So all in all, this shows that TD1473 at all 3 doses led to greater reductions in this disease surrogate biomarker than placebo. So similar to the biomarker data, we saw similar trends in histologic activity. On the left, you see a graph of the mean change from baseline plus and minus the standard deviation. And you can see that there is a trend for greater reductions in the Robarts histopathology index in the 20 and 270 milligram groups. The 80 milligram group did not show the same pattern, possibly related to the fact that the baseline biopsies in a few subjects had a very low histologic score, likely related to the fact that the biopsies were not done in an area of worse inflammation as was instructed. Now remember, this piece of data was seen at week 4, which is a short period of time after treatment started. And so we had anticipated very little movement in histologic activity with such a short duration of a treatment. And therefore, we're encouraged to see that at least there was a positive trend in the reduction in histologic activity at the 20 and 270 milligram groups. We then used the same batch of biopsies to measure on the right the amount of phosphorylated or activated STAT3. And as you may remember, STAT3 is right downstream of JAK. And therefore, what we're measuring here is the level of JAK signaling. So a negative a change would mean that there is JAK inhibition. And here you can see that there is a trend to show that there is JAK inhibition at the 20 and 270 milligram groups, with the 80 milligram data difficult to interpret given the biopsies may not have been done at the area of worse inflammation. So taking the data on the left and the right, one could say that the reduction in disease activity seen on the left is potentially related to the JAK inhibition by TD1473, at least at the 20 and 270 milligram doses. And last but most importantly were our clinical observations. On the left, you see that there is a trend for higher proportions of subjects achieving clinical response defined by the total Mayo score reduction, and mucosal healing, which is defined by an endoscopic subscore of 0 or 1. Now, this is with 4 weeks of treatment. On the right, you can see, consistent with the data on the left, that there was a trend of higher rates of subjects who had improvement in endoscopy or rectal bleeding at week 4. I don't have time to show the data, but the rectal bleeding improvement, actually, there was higher rates numerically for all 3 doses compared to placebo at week 2. So in summary, with the totality of data from this very early exploratory study that consisted of a confirmation of gut selectivity previously seen in animal models and positive data regarding clinical outcomes, histologic activity, and biomarkers, gave us the confidence to progress TD1473 further in development with a current phase 2 study in Crohn's disease and a phase 2/3 program in ulcerative colitis. In conclusion, the story of TD1473 could serve as an example of how an early exploratory study can enable early decisions to progress in clinical development a new molecule using totality of data. Thank you for your attention.
Great. Well, Deanna, thank you for a very nice overview of the early development of your program. And we'll move now on to our final speaker before the panel discussion. So I'd like to introduce our third speaker, my colleague, Dr. Niels van der Kastelen, who's Director of Precision Medicine here at Alimentiv. Niels has a bachelor degree in pharmaceutical sciences and a master's in drug design and development from Leuven. And he did his PhD in pharmaceutical sciences entitled Optimizing Anti-TNF Therapy in IBD Patients Towards Personalized Dosing in Leuven. He's an assistant professor of medicine at the University of California, San Diego, and he's director of precision medicine at Alimentiv. And he oversees the clinical pharmacology and translational research program that really focuses on development, validation, and implementation of clinical and translational tools to use in early drug development programs, both for sponsors and investigator-initiated trials, with a focus around PK and PD relationships. Niels is going to give an overview of the Alimentiv Translational Research Consortium and how it might help to drive innovation with methods development and validation, and also operationalize some of these things into complex clinical trials. So welcome, Niels, and thank you.
Thank you, Viv, for the kind introduction. So as we heard from the prior speakers, precision medicine can be implemented at every stage of drug development, from the preclinical stage all the way to phase 4. And typically, a systematic approach is taken where one first performs a comprehensive review of the existing data and the literature to inform the biomarker plan and incorporate that into the study plans. So from a clinical pharmacology perspective, one needs to define a strategy in line with the intended pharmacological profile. For example, the approach would be different for a systematically active compound versus a gut-targeted one, like we just heard from Dr. Nguyen. Also, population pharmacokinetic and pharmacodynamic modeling can help to understand variability in response between patients and guide dose selection or optimization. So from a translational analysis point of view, once the biomarker strategy is defined, a plan is put in place to outline each step from sample procurement all the way to sample analysis, which also encompasses choosing the right assays, which sometimes require optimization. And then finally, on the far right, the critical part, as we heard from Dr. Vermeire as well, is the integration of data from different sources, including the patient demographics, pharmacokinetics, biomarkers, or omics data, and the outcomes. And this compiled data set then allows for comprehensive data analysis and interpretation of results. And key aspects of that important step are the bioinformatics, the biostatistics, and pathway analyses to allow for interpretation of results in the right context. So precision medicine at Alimentiv is being operationalized through a consortium of 8 world-renowned clinical translational research centers. The Translational Research Consortium was established in 2015 and focuses on 2 research streams. One is the development, optimization, and standardization of research methods to allow for conducting state-of-the-art translational research. And the other is the application of these methods in multi-center clinical trial setting, or what is often referred to as true bench-to-bedside research. And so in the next few slides, I will highlight some of the past, current, and future focus points of the consortium. So flow cytometry with quantification of immune cell subsets is an extremely powerful technique in immunology. It's typically carried out in one lab close to the clinic, but that makes it difficult to implement in a multi-center clinical trial setting. Therefore, the consortium focused on a large standardization experiment where peripheral blood mononuclear cells were prepared at one center and then shipped to all other centers to conduct permeabilization and staining to identify areas of variability in results. And so our study found that when a standardized protocol is used, the main source of variability actually comes from the gating procedure. And so by conducting gating of cells centrally, we were able to reduce the variability to less than 25%, which is deemed to be acceptable for biomarker studies. And so these findings support the use of flow cytometry in global multicenter biomarker trials and lay the groundwork to conduct flow cytometry on colonic tissue biopsies. However, the main issue with conducting flow cytometry on colonic tissue biopsies is that you need fresh tissue. And this is very difficult to operationalize across a multicenter clinical trial where some sites may have very limited capabilities for sample processing. We have therefore, with the consortium, developed a cryopreservation protocol that allows sites to take the biopsy during the endoscopic procedure and immerse in a cryopreservation buffer that can then be stored at -80 and shipped on dry ice to a central laboratory that performs the cell extraction and downstream flow cytometry. The results of this approach and our studies showed an overall decrease in cell viability, but consistency in cell proportions. So here you can see the flow cytometry pipeline for blood and tissue, where the samples are being collected at the clinical site first, and then where the downstream storage and processing occurs. Currently, blood still requires on-site isolation, whereas biopsies can be cryopreserved and then shipped to a central location to conduct flow cytometry as well as central gating. So it is not well known what the impact is of location of biopsies on downstream biomarker and histologic disease activity assessments, as we also heard from the prior presentation. We conducted a study in 51 Crohn's disease patients with active ulceration on endoscopy, and biopsies were obtained at 3 locations, either the ulcer edge, 1 forceps away, or 2 forceps away from the ulcer. And what we found was that biopsies procured from the edge of the ulcer yielded the highest histologic disease activity score and mRNA expression. Furthermore, when the biopsies were taken consistently at the ulcer edge, no difference was observed between the ileum and colon in the measures that were investigated. And so these results further underscore the importance of biopsy location and standardization across studies, both for biomarkers as well as for histologic disease activity. Now, once samples are procured at the clinical site, a rigorous process is followed to ensure high quality quantitative data. And here you can see the general biomarker pipeline, where first input is provided on sample procurement, which is then included in the lab manual and informs the development of the collection kits that are then being sent out to the central lab. The clinical sites collect the samples in the appropriate recipients and will ship these back to central lab for biostorage. Next, these samples get distributed among the specialty translational labs of the consortium according to the analyses that are being performed. And all the generated data from the labs is then being collected and stored centrally at Alimentiv to allow for data integration, analysis, and interpretation before results, together with the integrated data set as well as the raw data, is supplied back to the sponsor. So from a data analysis point of view, clinical pharmacology can be implemented at all phases of clinical development to better understand between subject variability and variability within one subject over time in pharmacokinetics and pharmacodynamics. In particular, for gut-targeted compounds like the one discussed by Dr. Nguyen, requires a comprehensive multi-compartment modeling approach to understand the kinetics between systemic and tissue drug exposure and the relationship thereof with efficacy and safety. This may support dose selection or identification of subgroups of patients who may benefit from dose optimization or who may be better candidates to switch to a drug with a different mechanism of action. This too can help make a clinical trial more efficient, not only by selecting the right patient population, but also by targeting the right drug exposure at the right location. So to further address the unmet medical need in inflammatory bowel disease, our consortium takes a systematic approach to conducting real-world observational biomarker studies that may support optimization of therapies that are on the market. There are 3 steps taken every year to focus on one particular drug class with a particular mechanism of action. And for example, in 2019, these were the JAK inhibitors, as also Dr. Diana Nguyen talked about. So firstly, we organize a symposium that brings together expert clinicians, scientists, pharmacologists, clinical trialists, and industry to review and discuss existing clinical, pharmacological, and translational evidence of these compounds that are on the market or in development. Secondly, these findings and discussions are summarized and published in a literature review that outlines the research gaps as well. And thirdly, a prospective observational study is designed to address these identified research gaps. So these real-world prospective observational studies are conducted according to the same standards as a clinical trial, and the collected data could be used potentially for a label change in line with recent developments of regulators being more open to the accepting of real-world data. This year, we focused on S1P modulators, and we are already preparing for the 2021 symposium, which will be focusing on the IL-23 pathway. And please don't hesitate to reach out later if this is something that you would be interested in attending or contributing to. So finally, to put this all into perspective, I've outlined here on the right the precision medicine life cycle that is operationalized through the Alimentiv Translational Research Consortium. You can appreciate how method development and optimization helps to enable observational biomarker studies that will provide further insights that can then be applied to new compounds or clinical trials to make early drug development more efficient. Overall, this is how we see the process of precision medicine for accelerating drug development by fostering public-private partnerships between academia and industry. And with that, I would like to end my talk with a quote from the FDA that success of personalized medicine fundamentally depends on the development of biomarkers and diagnostic tests that can be used to accurately identify the patient population. I would like to thank you for your attention and happy to take any questions during the panel discussion.
Okay, wonderful. Thank you, Niels, for that overview. And I'd like to thank all of our presenters today. We'll move on to our panel discussion now. We have about 10 minutes for some questions. So I'm going to jump straight into the first question, actually, that's come through the panel box, which aligns with a question I had too. And this is a question for Séverine. The question is, please could you expand on your point regarding harmonization of clinical definitions? And why will that be important for biomarker discovery and validation? And how might we address this collaboratively as a clinical community?
Yeah. So if we're discussing, for instance, primary non-response to drugs, we're seeing many different definitions being used. So some will use a pure clinical definition based on PRO, others will use a definition based on CDAI. Okay, you can say it's almost the same, but still there are differences. I think more and more we think that a true primary non-responder should be defined not only by clinical absence of any improvement, but also no mucosal improvement. Plus, on top of that, we think that also pharmacokinetics should be added, and I think adequate drug exposure should be, or at least it should be corrected for drug exposure as well. So I think there, when we look at the individual data sets that are being compared, the definitions for what we call a primary non-responder are not always the same. And that should be, I think, an easy one to get a consensus around. The same thing for a loss of response. How do you define a loss of response? Is it reappearance of symptoms with adequate drug exposure? Do you take that into account or not? All of these things.
And thank you. And could I just maybe expand a little bit on that question? I mean, one of the things that you very nicely highlighted is to move this field forwards. There's going to be power in merging biorepositories, and there's lots of global initiatives around that. And my understanding again is that many of these are based on biobanks, which sometimes are built from the ground up without necessarily predefined hypotheses. So, given that there'll be different types of omics data at different time points and different characterizations of patients, do you foresee challenges in really merging all these clinical data sets and repositories? I mean, do these have to be done prospectively again with very standardized, or I mean, do you see that as a challenge?
Of course, and in the perfect world, you want the whole world just to sit together first and to come up with the same definitions, the same way of handling samples, storing samples, doing the analysis, absolutely. But I think in the meanwhile, if we really want to advance, we have to start working with the samples we're having with the different centers. I think the consortium that Niels showed was a very good example of a nice collaboration, which is still ongoing, as you know, where there are certainly differences between the centers, but over the years, I think we have also aligned ourselves more and more. Also, time points, when do we evaluate? So yes, in the perfect world, you would like to start building this from scratch, and everybody in the same way. But this is not going to be feasible, I think, for the next couple of years. We could only try and harmonize as much as possible definitions, time points of sampling, and I think that already should help.
Great, thank you. I might move on to a question to Niels, if that's okay. So I think, Niels, so far, I mean, in development programs, we use fairly rudimentary biomarkers, fecal calprotectin, CRP, particularly in Crohn's disease. And do you see inclusion of very tissue and gut-specific biomarkers as an essential component of drug development programs, not just for the gut-selective agents, but more generally?
Yeah, it's a great question, Vip, and I think CRP and fecal cal will remain important. I think there's these are the biomarkers with the majority of evidence. So probably if you think of regulatory ready biomarkers, CRP and fecal cal are the ones that have the most evidence and are closest to getting that status. That said, I do think there is a role for biomarkers earlier on in the clinical development program. So we know that there is substantial heterogeneity in our clinical endpoints. So that's not only clinical symptoms, but also endoscopy and to some extent histopathology. And I think that's where we can use biomarkers to get a readout that is objective and give us an idea of whether we're hitting the targets and whether we're hitting it hard enough. So I think these biomarkers can help with drug development, confirming that there is target engagement, but also help then with dose selection, or who knows, potentially even identify a subgroup of patients who are more likely to responding to the therapy. And it's the sooner that is being initiated, sometimes even in a preclinical phase where some of these biomarkers are tested in animals and then translated into humans, I think the opportunity is potentially even to get to companion diagnostic biomarker, which is still something that remains to be done in the field of inflammatory bowel disease. So I think great opportunity there that lies ahead.
Great, thanks, Niels. And another question has come in, Séverine, maybe I could ask to you, but anybody could take this as well. Of the development programs of drugs that we have right now, we seem to have hit a ceiling in terms of efficacy rates, 50% at 52 weeks. And you touched on the point of we enroll all Crohn's disease into the trial, a very heterogeneous group, and you highlighted perianal, ileal predominant, chronic dominant. The beyond clinical phenotype, how else might we enrich patients going into studies if we were going to look for potentially more homogeneous Crohn's disease, whatever that? Could you share your thoughts on that?
Yeah, so I think more molecular subgrouping of patients is what is needed. And I don't necessarily think this should be done on a huge amount of patients, but I think if and these collections are ongoing, if individual integration on an individual level is done on a sufficient number of patients, and I'm not a biostatistician, so I don't know how many of these patients you would need with the integration of the same omics layers. But I'm sure that we can start at least to identify some subgroups, molecular subgroups of patients. And I think for me, then the next step should be that we no longer just allow all Crohn's patients with inflammation into a trial, regardless of disease location, disease duration, previous treatment, smoking or not, but that we really focus on some subgroups that are defined by some biomarkers. This would be already a big step ahead, I think, more homogeneous patient population. The other thing for the ceiling too is that the drugs are all anti-inflammatory, but I think we really, really need to start working also on on fibrosis, on the microbiome, on the barrier, and I think we need to combine that with anti-inflammatory agents. The disease is not just inflammation.
Thank you. Deanna, there's a quick question that's come in for you in the chat box here. So, question is: were Tregs or Th17 evaluated in the clonic biopsies after day 28 of TD1473 treatment?
That's a great question. Ideally, we would love to with a bigger sample size, but we only have roughly 10 per group. We did do some RNA analysis, and you can see more details in the JCC paper, but it was very difficult to then have to isolate fresh cells to look at Treg populations and Th17 populations. But that would be an ideal thing to do with a bigger study with a bigger sample size. However, as Niels pointed out, the feasibility of doing something like that in a bigger, more global study would be the operational aspects of collecting samples and then isolating fresh cells from those.
Great, thank you, Deanna. And maybe I could ask you to maybe take the last question, Deanna. There was a really nice presentation you gave and a phenomenal experience that you've outlined of the development program the last few years, and it was great to see publication of that as well. Could you share some high-level lessons learned through this? Because I'm sure there were it wasn't straight plain sailing throughout that whole program. Could you share any high-level lessons that you learned from that program?
Sure. I think for a small exploratory study like this, which is not powered for any of the usual outcomes in a UC trial, you have to be creative and think of how to look at the data with the mindset of totality of data, since you're not expecting any statistically significant changes in whatever usual outcome you expect. So using totality of data is very important in a small study like this. And then, as Niels pointed out, I think the use of biomarkers as exploratory endpoints in these small early studies is really crucial. And whether it's blood or tissue biomarkers, these are really key to show target engagement, to show that you're having some effect, even if you don't see the significant changes in your clinical outcomes. And using biomarkers often means that you have to think about how to optimize the use of the biopsies you get, how to use it maximally with a small number of biopsies. And it's very important to instruct the sites how to collect and how to process these samples in a way that they can understand and remember, but still preserve the tissue for RNA and protein analysis. So in our small study, I and another scientist actually personally trained every site how to do this. So it's possible with a small study, but a little bit more challenging with a global study.
Great, thank you for those that outline. I think we're hearing a lot about standardization here, and I think you've highlighted that was feasible in a small program, but how do we roll that out to a 500 site IBD trial is more challenging. Great. Well, look, we're past the hour, and in the interest of time, I think we're going to wrap up here. I'd just like to really thank all of our speakers for all the time and effort that goes into these presentations, because many hours of prep goes into them. They're really a wonderful overview of this topic. So thank you again for your time. Thank you for the many hundred people who spent their time with us today. If there are other questions, please feel free to email them in, and we will endeavour to answer the questions offline. So again, thank you, everybody, and please look out for our next live stream event, which will be coming over the next 3 months, and further details to follow. So thank you, good evening, good morning, and good night, for depending on which part of the world you're calling in. Thank you.